This slide exposes one of the most dangerous gaps in 21st century medicine. We know only around 300 virus species capable of infecting humans, and even with this relatively small kind of group, effective virus-specific therapies exist for only a limited number. For many viral diseases, medicine still depends primarily on supporting care, or treatments develop only after an outbreak has already begun. Beyond this known landscape lies an immense biological reservoir. An estimated 1.67 million undiscovered viruses of mammals and birds. Hundreds of thousands may have a capacity to cross the species barrier. We cannot predict which virus will emerge next or how quickly it may spread. But waiting for the next crisis is not a strategy. Humanity is still trying to control an accelerating viral threat with a development process that is too slow, too expensive, and too dependent on exhaustive trial and error. In 21st century, this approach must be fundamentally redesigned. We cannot continue responding to every emerging virus by rebuilding the research process from the beginning. We need a modern antiviral infrastructure that integrates genomic data, structural biology, molecular modeling, predicting software, and experimental evidence into one continuous decision support system. Do you see an elephant in the room? Perhaps not, because most of us prefer to believe that we are safe in the major centers of modern civilization, surrounded by us, by advanced hospitals, vaccination programs, and high sanitary standards. We assume that modern medicine will somehow protect us, even from viruses for which no effective treatment exists. We behave as though a pandemic is someone else's problems until it reaches our own cities, our own families, and our own bodies. I sometimes joke that my immune system is so powerful, that is, it occasionally attacks its own owner. Surely it can handle with the viruses as well, at least while I'm still relatively young and healthy. So why should I make a video about the alarming state of antiviral therapy worldwide? Why worry now? Perhaps I should wait until I am, when older and weaker and seriously ill. Waiting until a viral threat becomes personal is not a preparedness. It's a denial, and it's a profound irritational. Instead of committing resources to 100 possibility in vitro experiment, the BinomLabs technology platform can help researchers identify and prioritize approximately 30 of the most promising candidates for experimental validation. The BinomLabs introduce a new research era and development approach designed to transform antiviral research planning. We do not replace laboratory research. It makes laboratory research more focused, strategic, and efficient. By scientific prognosis of experimental data, we can rank targets, compounds, antibodies, and molecular modifications. Before testing begins, we help researchers' team design a more effective experimental roadmap, reduce unnecessary trial and error, and concentrate laboratory capacity on the candidates with the strongest predicted potential. The next era of antiviral research must begin before the next pandemic. The BinomLabs provide an AI modern technology to help make that transition possible, turning large experimental spaces into focused, evidence-guided research programs. Every experimental biochemist knows how fragile a biological signal can be. A small change in buffer composition, ionic strength, temperature, or incubation time can weaken, amplify, or even reverse the observed effect. A signal detection in one cell line may disappear in another. Tissue-based and in vivo studies may produce effects of different strengths or results that appear to contradict one another. The experiment can no longer be described as simply positive and negative. Researchers are left with the data that are difficult to interpret, explain, and defend, and with no clear answer about what should be tested next. And this is exactly where the BinomLabs comes in. We connect these conflicting signals with molecular interactions, binding affinity, biochemical pathways, and possible reaction redirection. We help explain why the effect may change between conditions and provide a prioritized list of targets, controls, and follow-up experiments capable of resolving the uncertainty. No experimental left behind, no result lost in the fog of uncertainty. The BinomLabs turning conflict biochemical signals into clear experimental decisions. Thank you. We already possess the scientific knowledge, experimental capabilities, molecular data, and computational technology required to begin. What we still need is the determination to use them before the next crisis reaches us personally. Thank you very much.